Hello, I'm Veronica Bobskill. I work in Dr. Lover's lab as a seasonal surveillance technician manager, and these are vegetation analyses at long-term tick surveillance sites. So vegetation is largely the way that ticks interface with their hosts. Understanding more about vegetation may teach us more about their habitats and their life cycles. So we have three main goals. One, to quantify the interannual variability at a lot of different ecological sites. Two, to create a protocol that's comparable to collaborators. And also to conduct these analyses and our plot placement. So our typical tick collection plots are 40 by 40 meters, which is pretty huge. For this vegetation analysis, we only sampled the vegetation at a subplot every 20 meters, which, as you see here, these little blue blocks are the subplots, and they are 3 by 3 meters, comparable to what is being done in Vermont. And we have a total of 8 subplots per tick surveillance plot. And in the picture, there's James placing the plot, and we use a 3 by 3, a 3 meter PVC pipe to make it way easier to place the edges. Vegetation metrics. So we looked at leaf litter composition, invasive species density at a 0 to 3 scale, 0 being none present, 3 being pretty filled. As you can see in the pictures, we did the same thing with understory vegetation coverage. This included invasive and non-invasive plants. We looked for the tallest plant and shrub in the plot and quantified their height on a scale of 3. And we did presence and absence of select features, including mass species, tall grasses, and berry species, as all of these could lead to different hosts being around or a positive habitat feature for ticks. We also looked for leaf litter depth at 2 locations, one on each side of the subplot. We collected basal area and used hobo loggers to collect temperature and relative humidity, and searched for canopy coverage using a GRS densitometer, which that's the view through it. You kind of look through the crosshairs at a little point, and we looked at 8 different spots per plot. Takeaways. It's important to balance collecting qualitative and quantitative vegetation metrics when working with limited time. So if everything was quantitative, it wouldn't be possible to get this work done in a timely manner. And we also could not use the exact same sampling methods due to different habitat and, again, staffing. And it was really beneficial to create tools to make sampling simpler. And I have physical copies of this protocol that are happy to share with anybody if they'd like to look it over. I'd like to thank my field crew and collaborators and the Lover Lab